Hello, boys and girls, and welcome to today's lecture on non-Mendelian genetics. This means that today we're going to be talking about incomplete dominance, codominance, and epistasis. So, today's lecture, we're going to be focusing on right here with incomplete dominance, here with codominance, and here, which is a mixture of sex-linked dominance and codominance. So, first, what we're going to talk about right now is incomplete dominance. And this says that some alleles for a gene are not completely expressed, so are not completely dominant over the others. And this creates a partially masked phenotype. And this means that we get a intermediate phenotype. So here, for example, we have a red and a white, and they're going to make a purple child. Now, we can write this in a couple of different ways. We can write this as, in its normal way of big R, big R, big R, or little r, and then the heterozygote, of course, being big R, little r. And that's how we can go from this one. Or the other way we can write this is by going through it and using primes. So the first one will be R, big R, just like normal. The other one can be R prime, R prime. So R prime, R prime. And then the heterozygote being R and R prime. Now, the one they're using in this slide is R and R prime. Now, I'm going to go up here to the top, and we're going to do R, R prime, and then R and R prime. And so our first square, we go down and across, and we get two big R's. And in our second square, we get an R and an R prime. Let's like K real quick. And then in our this square right here, we get an R and an R prime. And then in our last square, we get two R primes, R prime, which then is the same right here. We have big R, 2, and this is the reason why they do it, is they want to show you that 2 of them are heterozygous. They get in big R and then an R prime. And then 1 that is 2R prime, which gives us the ratio of 1 to 2 to 1, not only for our genotypic, but also our phenotypic ratio. So our phenotypic ratio changes because we have that intermediate phenotype. So we have 1, big R, 2, R prime, and 1, R prime, and our phenotypic ratio becomes 1 red to 2 purple to 1 white. So our phenotypic ratio and our genotypic ratio become the same, at least when it comes to this example here. Now for this example, what they decided to use was they used superscripts. So they said C for color, and they said C with a big R is going to be a red phenotype. C with a W is going to give us a white phenotype. And so red plus white gives us a pink phenotype. And so then what we did was we crossed two pink flowers, and we got one red, two pink, and one white flower. So that's our phenotypic ratio. So our genotypic ratio would be 1 CR, CR, because you got to remember we're using what they gave us. 2 CR, CW, and 1 CW, CW. So we get a 1 to 2 to 1 ratio on both sides again. And remember, genotype is talking about the genes, and that means we're talking about the letters in particular, the alleles, what's in your genes. Where phenotype is talking about the physical traits, pH, physical trait that's expressed. So usually you can see it with your eyes. Not always though, because think about it this way. Think of a couple of examples of what you can't see with your eyes, but is still a phenotype, like diseases, for example. Now right here, we are seeing some intermediates. So here, these are golden retrievers. And we get the white goldens, the red goldens, and then of course the regular golds. And then here we have some palomino horses, which are also part of all incomplete dominance. Which means we get that third phenotype. Now, codominance, this means that we have two alleles that affect the phenotype in separate and distinguishable ways. So they are separate and distinguishable. They are both dominant. Neither allele can mask the other, and both are expressed, and they are not in an intermediate form. Keyword there being not in an intermediate form. So our example here is we have red flowers and white flowers, and they give us both red and white flowers. So there's no pink flowers there. The flowers have red and white in that flower. So for example, here we have Oreo cows right over here. And these are cows that you guys can see all the time. They're more common in South America than they are here. But you get the black face, the black butt, and the white middle, which is why we call them Oreo cows over here. Just so you know, Oreo cows, you can find these actually in parts of Northern California, but this is the picture that we're talking about when we say Oreo cows. Now, here's an example for you, and I want you all to write down this example and then come to class with it next time, because we're going to go over this one in class. So in cattle, a roan coat color, which is a mix of red and white hair, occurs in the heterozygous, and the offspring of red and white. When two roan cattle are crossed, the phenotypes of the progeny have the ratio of 1 red to 2 roan to 1 white. Which of the following crosses would produce the highest percentage of roan cattle? So which will produce the highest percentage of roan cattle? So I'm going to underline it again. So red, white, and roan is the heterozygote, okay? So roan is heterozygous. Now, knowing this, which of these are going to give you the highest percentage of roan cattle? Let's do one of them together. We're going to do D. So red is big R, and white is little r. So, oh, that's it. Roan, big R, little r, because we're doing D. So, big R, big R, big R, little r, big R, big R, big R, little r. So this gives us a 2 big R to 2 big R, little r, which means we get 2 red to 2 roan. Is that going to give us the highest percentage of roan cattle? I want you to do the others and figure out which one is going to give us the highest percentage of roan cattle. This right here is a picture of Scooby-Doo, but it does show codominance, and it shows it in the form of his fur. So right here, if you look at his fur, you get the brown fur and these spots of black fur. So that gives us a codominance. So right here, we're talking about multiple alleles. Now, multiple alleles means that we have multiple copies of alleles for the same gene. So for example, right here, we have O, which is a recessive genotype, which means you need 2 little i's for O. Now, people who have the O blood type usually do not make any surface antigens, and that's why they're O. Where if you're A, you're going to make a little antigen that says, hey, I'm an A blood cell. Now, the biggest thing about this is you create an anti-B antibody. And this says, hey, if I see this B blood, my body's going to clump because this is not normal, this is not my blood, and I need to get rid of it. Same thing over here with a B blood cell. They're going to create anti-A antibodies. And it's the same thing. It just says, hey, if I see this blood, this is not normal. I need to clump and attack because this is not my blood. And a person who is AB, which means they have both the A and the B allele, they are codominant, does not have either of them, does not have either antibody because they have both surface antigens. And those surface antigens say, hey, I'm an A and a B blood cell. And if we made antibodies against either one of those, the body's going to start clumping every time it sees any blood, which isn't good. Now, because we have these antibodies, this tells us who we can and cannot donate to. For example, a person who has O blood can only donate to, or can only accept from O people. And the reason for that is O people don't have any surface antigens, which means those anti-A and anti-B antibodies won't be acting up. Where a person who has A blood can accept from an A person because, hey, it's the same blood group, or an O, and that's because they don't have any surface antigens that says, hey, I'm a B. Same for B. They can accept from an O person or a B person because they don't have any surface antigens that say I'm A. Where an AB person can accept from everybody. So the phenotypes are controlled by more than one allele. This is multiple alleles, mind you. And blood types are not only codominant, but they are regulated by 3 separate genes. So for ABO, which is one of the 32 different types, we have different surface antigens. Now you can be A, so here's the genotypes that we use, IA, IB, and IO, or you can also use little I, that's fine. And these are the 3 alleles that determine blood type. Now, type A means that you have the A surface antigen, and this is denoted by IA. Type B says you have the B surface antigen, and this means that you have IB. And type O says that you have neither A nor B surface antigens, and this is denoted by IO or little I. And here are our 4 blood groups. You can be A, B, AB, which is the universal recipient because it can accept from everyone, and O, which is our universal donor, which means it can donate to everyone. Now, a person can receive blood only when the donor's blood type does not contain the surface antigen that the recipient does not have. So, if you have B blood, you don't have an A antigen, so the body you cannot accept from them. This is because of the antibodies that we make, which will attack any foreign surface protein. So AB, because it has A and B, does not have any any antibodies. So a person who is AB can only donate to another AB person. This is known as the universal recipient because they can accept from everyone, but they can only donate to themselves. Same thing for type O, they're a universal donor because they can donate to everyone. They have no surface antigens, but they can only receive from type O. Now, the other factor that we have is positive and negative. This is the second blood group of the 32. So we've now just hit 2 of 32 blood groups, and we're not going to talk anymore after this. We're only doing the 2. But you're either Rh positive or Rh negative. And how we studied this was we actually studied this in rhesus macaques, who have very similar blood groups that we do. And this is based off of a dominant and recessive mechanism. So this is Mendelian. This is based off of Mendelian genetics right here. Oh, that changed that. Okay. Now, this is not usually a problem, except when it comes to pregnancy. So it's not usually a problem except with pregnancy. And the reason for that is if we have an Rh negative mother and she's carrying an Rh positive fetus, she can develop antibodies that can attack and destroy fetal blood. Now, what I'm meaning when I say this is to have a negative blood type, you must be negative. You must get both from your parent. And what this is very similar to O. It's recessive. So here we have an O person, and they don't have any they don't have any flags that says anything else. And it's the same with negative blood. They don't have a flag that says, "Hey, I'm a positive blood cell." So here we're going to have our positive person. They have a little flag and it says, "Hey, I'm positive." O doesn't have that. So because of that, the Rh negative parent, when after the first pregnancy, there's usually hemorrhaging. And that hemorrhaging makes it to where the blood flow intermingles, and then the mom is now sensitized. And if she sees any positive blood anywhere, her body is going to attack it. And this is why it's problematic for the second and third pregnancies, because then it's detectable and the body's going to try to destroy the fetal blood. Because we've learned from this, we now know we can detect it and we can treat it. It's called RhoGAM, the thing we used to treat it with, and it's usually given during the second trimester. Pleiotropy. This means that most genes have a multiple phenotypic effect. So this means that the ability of a gene to affect an organism in many ways. So here we have sickle cell anemia, and it is a Mendelian genetic trait. So it means you have to have both little s alleles to get it. You're big S, little s, you're heterozygous, which means you still have normal cells. So here we have our normal cells, and here are sickle cells. And what ends up happening with sickle cell anemia is if the oxygen content is low, the red blood cell will become sickle-like and will freeze in that sickle shape. And what ends up happening is we can have multiple phenotypic effects from this. We can get the breakdown of red blood cells, which can lead to physical weakness, anemia, and heart failure, which anemia can lead to impaired mental function. We can go to the clumping and clogging of blood vessels, which can lead to pain and fever and brain damage, which that can lead to impaired mental function and paralysis. We can get damage to the other organs, which can lead to pneumonia and other infections, kidney failure, or we can get the accumulation of sickle cells in the spleen, which can lead to spleen damage. So these are all things that can happen, and this is just multiple phenotypic effects coming from just one gene. Now, epistasis occurs when a gene at one locus can influence or alter a gene at another locus. So here we have C is color, and this must be dominant for pigment, which is B, to be expressed. Okay, so if color is dominant, we can have the other allele be expressed, but if it's recessive, then it's not going to show. So here we go, we have two heterozygous mice, and we get not, since 9 showed the dominant trait in both traits, remember we would have normally a 9:3:1 ratio, but this is epistasis, meaning that C is going to affect how B is done. So right now we have 9, and 9 are showing that black color. Okay, 9 are showing that black color. 1, 2, 3, 4, 5, 6, 7, 8, 9. There we go. 3 are going to show brown color because that pigment that we were looking for is recessive. But the only other thing that we have here is now the other 3 that would be positive. The other 3 that would be for the other 3 that would show the dominant trait in pigment has the recessive trait in color, and color affects pigment. It says, oh look, I'm recessive, so you need to not be on anyway. So we get 3 white mice, and then this one's a white mouse because it's both. So instead of 9:3:1, we now have 9:3:4 for our phenotypic ratio. So we can count it out. Because the little c's right here, this affects how the B's are done. So the little c's affect how B is expressed. That's epistasis, how one gene can affect another locus and how it is expressed. Polygenetic inheritance, this says that we have multiple additive effects from 2 or more genes, and this is usually controlled by at least 3 separate genes. Skin tone, height, these are all polygenetic. right here you see we have 2 heterozygous people, and we can get a nice bell curve of the different children possibilities. And that's just from 3. Like, if we had more, that would be even more of a bell curve. So polygenetic inheritance, it just says that we have an additive effect from 2 or more genes. Pigmentation is controlled by at least 3. Here is another one. This one is height. So height in particular, and they took a picture right here is from a school where they separated everybody by their height, and you can see that this is a bell curve here, and you get this bell curve right here as well. Like, we're not just tall or short, we have a wide range of continuous values. All right, so next time we're going to be talking about sex-linked traits. Next time we're going to be talking about how the environment can affect your genotype and how it can affect your phenotype. And we're going to be talking about what is the current average phenotype of America. All right, have a good day.